morning, everybody. My name is Samantha, and I'm a fifth-year PhD student in Dr. David Resnick's lab here at UCR. And so I do a lot of research about how mothers interact with their offspring, and the fish that I use for it is pretty interesting. So I'll introduce my talk, Can Mothers Differentially Allocate Resources to Offspring Sired by Different Males? So, why am I showing you pictures of deer if I'm talking about fish? Well, I think that this really illustrates my research pretty well, which shows that mothers need to provision their offspring. They need to nurse them in the case of deer, but when does she stop, or when does she need to stop? Clearly, in the case over here, we have a fully adult deer nursing on a fully adult deer, which probably has taken it a little bit too far. So that's where my research comes in. Who controls offspring resource allocation in placental organisms? Well, we can look at it from two different perspectives. The first perspective is from the mother, and evolutionarily she's going to be predicted to allocate resources evenly across all of her offspring to give all of her offspring the best chance of survival. Now, conversely, or as we see here, perhaps to her chagrin, but conversely she's not the only player here. There's also the father and the embryo, which may be predicted to have some different ideas of how much their offspring should get, especially in the case of cats and many organisms that mate multiply with many different males, who actually controls how much each offspring gets. So the father / the embryo, the father acting through the embryo, we could say, is predicted to act selfishly at the expense of the siblings and the mother. So this is predicted to result in a tug of war over resources. Now, when I talk about placental organisms, we might think of placental organisms, right? Lots of mammals. But there are a fish family that I work with called the Poeciliidae, and in this family we've had the evolution of a placenta-like organ. It's not exactly the same because it's not from the same origin, but it's a very similar organ, and we've had this evolve at least 9 times in this family. And so with this evolution of the placenta in these fish, we can start to ask questions about placenta evolution and also how placentas work on a scale that makes it a lot easier to study because we're working with smaller organisms that have faster gestation and do many of the same things that mammals do. So the fish that I'm working with in my study is Heterandria formosa, and I have some of them right here as well. These are, as you can see, they're quite small fish. I'm not sure how easy it is to see them, but the mothers and fathers, they only get to about 20 millimeters in size, which you can think of as about the size of your pinky finger. So they're not very big, but they are easy to study, they're easy to keep, and my study tanks are actually these tanks. So I keep fish in these tanks for their whole lives, and they're perfectly happy in that environment because in nature, they live in very shallow water with dense vegetation for the most part. And even though you see, these big habitats, they're only living in this very small range near the shore. So easy to catch, easy to see, easy to keep, great study organisms.
And where is Lake Jackson?
Oh, that's a great question. Sorry. So I'm originally from Florida. I did a lot of work with these organisms at Florida State University. And so these are Florida fish. They expand all the way into like Louisiana, a little bit into Georgia, maybe South Carolina. So these are Florida lakes. As you might see, these are very much not California habitats. So Wazuzza River and Lake Jackson are two populations of Heterandria in Florida that are located about 20 miles from each other, but they're genetically distinct populations. And what's really interesting about the ecology of these areas is that in lakes and rivers, they are quite different, even though they're both freshwater habitats. So Lake Jackson is what I call an SO population. What I mean by SO is that they produce small offspring. So they tend to have many offspring that are smaller. And the reason for this is that in those, shallow lake habitats, there are lots of predators. And so the mothers are going to produce as many offspring as they can to give them the best shot of survival. Conversely, Wazuzza River I call a LO population, or a large offspring producing population. These females have fewer offspring that are much larger. And the reason for this, they have fewer predators, so their population densities are a lot higher. Lots of fish means lots of competition, means that you want to produce really big offspring that are going to have the best chance at outcompeting their conspecifics, or their fellow species of the same species. Fellow members of the same species. So one thing we can do with this small offspring, large offspring aspect is we can look at how mothers provision their offspring based on whether the father is from a small offspring or a large offspring population. So on this x-axis here, I have the maternal population, and on the y-axis, I have dry mass of surviving offspring. Now, this is not my study. This study was done by Schrader and Travis in 2008, and what they found is that, when you have your sire population of SO or LO, open squares or solid squares, we find that small offspring females, when mated to small offspring males, produce small offspring. That makes sense, it's the same cross. When small offspring females are instead mated to large offspring males, they produce larger offspring, right? So we see that the large offspring males have some pull over the females. They can make the females make bigger babies for them. However, when large offspring females are mated to either kind of male, they produce pretty big offspring. So we see that these females are just provisioning a certain amount of resources, whereas these females produce different offspring sizes based on who they're mated to, which is really interesting. And I think it really begs the question of what happens when you mix the two? What happens when you give females the potential for choice, males the potential for competition, all within the same female? So that's exactly what I did. So my work involves artificial insemination, in which I actually gather sperm from males from both small and large offspring males, and I mix it together, and then I inseminate the females with it. So I use a controlled amount of sperm from each male, and then inseminate females, wait, collect their offspring. So that brings us to this question of when females are mated to males from their own or a genetically distinct population, do females allocate resources differently to each of their offspring? So we have all of these babies here. Who belongs to who? And so the follow-up question here is there a bias in paternity? Who's siring the big offspring and the small offspring? So let's move on to my predictions. So in this case, you want to always have controls, of course, as we're learning in this course. So in my case, I have 2 large offspring and 2 small offspring males as sort of my A and B treatments, or A and B controls. And then my third treatment is when you mix both of them, 1 LO male, 1 SO male, what happens? So for my predictions, with large offspring females, we've been given evidence before that large offspring females are going to produce large offspring regardless of who they're mated to. But for small offspring females, we know that if they are mated, so inter by the way, inter meaning different, so a small offspring female mated to a large offspring male, inter meaning producing large babies, as we saw from the previous study. The control in this case, small to small, we'd expect small. But what about when we mix it? Well, we kind of have a 2-part hypothesis here. We could either see intermediate sized offspring in that there are some large, some small, so the average is going to be in the middle. Or we could see some sort of bias. And so this is what that intermediate allocation would look like. I'm going to orient you here based on our cross, a small offspring female in this case, mated to 2 large offspring males, 2 small offspring males, large and small, and then the mixed being mixed and intermediate size. Or we could see a skewed result. And in this case, additive means the adding of the 2 types, small and large being the middle ground, and then the skewed being non-additive, meaning that maybe there's some bias. And so what would bias look like? Bias would look like, well, the average offspring size in this small offspring plus large offspring cross would be relatively small or relatively large. So, and then as for paternity, we would expect random paternity in the LO and SO. In this species, something I should have mentioned before, is that they don't have courtship. Unlike guppies, which you may be familiar with, this species is called coercive mating, meaning that males and females just mate with each other. There's nothing about a particular male that would visually appeal to a particular female. Now, that doesn't rule out post-copulation sperm competition, but for the most part, there is random mating in either of the controlled cases. But as far as our predicted paternity, for the LO and SO females, when they're mated to both kinds of males, LO with both, we would predict maybe a bias towards LO, that they would prefer males from their own population, or that their own males are better at fertilizing their own females' eggs. And then for SO, we would predict the same, that there would be a bias towards SO. But what's great is that if you're kind of thinking about, well, what's our module going to be, this is the part where the module comes in, and we'll talk about that in a little bit. Okay, but what I will show you is I'll show you the results for size. So we're not going to do that in the module, but I'll give you an idea of what we saw. So this is an example here, and I also have tubes of them. So these, by the way, are what a fully mature female looks like and a fully mature offspring that was given birth to. They're quite small, as you can see, but this is a better picture of what that female and her offspring might look like. We have a female that weighs around 350 milligrams, and then her offspring at birth weigh between 1 and 5 milligrams. So already we see that there's quite a large range in offspring sizes. So we're going to look at our LO females first. So large offspring females, these are the ones that tend to produce large offspring regardless of who they're mated to. And even though the results here with the cross on the x-axis and the weight in grams on the y-axis show that there does look to be some differences, these error bars here give us an idea that they're not statistically significant from each other. In my data, I analyzed it using generalized linear mixed models, which are similar to ANOVA tests that you might have used in your classes before. And what we find is that the p-value here is about 0.48, which you might recall that a p-value of less than 0.05 means that your data is statistically significant, or that there's a less than 5% chance that the result you see is due to chance. So in this case, you could kind of read it as there's about a 48% chance that this result here is due to chance, and so we accept that as being not different from each other. What this is to say is that large offspring females produce relatively large offspring, regardless of whether they're mated to their own males, different males, or a mix. That doesn't tell us anything about paternity, though, which we'll get into later. So now let's look at our small offspring females. Well, the first thing I'm going to do is I'm going to show you the difference between the large offspring and the small offspring only crosses. They produce large offspring when mated to large offspring males. They produce small offspring when mated to small offspring males. This is the result that we already knew about. So what happens when we mate them to both? Well, interestingly, we see support for that secondary hypothesis that I was talking about, that there's a skew in offspring size. The offspring are relatively small in this third cross. When we mate small offspring females to one of each kind of male, they produce relatively small offspring. So what's cool about this is this means that either they're preferring those small offspring males, well actually that is the main idea that we would have, or there's something that happens with those large offspring males. Maybe the females are able to give them fewer offsprings or fewer resources when the sperm is actually mixed together and you have offspring of both origins. So next we want to talk about our paternity results. These are hypothetical paternity results here, in which if we have a small offspring female, maybe the smaller offspring are from the small offspring male, larger from larger. We're not sure. That's what we're going to find out. So this is for you to find out, and how are we going to do that? Well, after we've done our experiment, we've weighed our offspring, we've put them into 100% ethanol to preserve their DNA well, we are going to do a multiplex PCR reaction and then look at a couple microsatellite loci, genotype our individuals, and use that to deduce who sired who within a brood. So what's a multiplex PCR? We've talked about PCR already in this class, and multiplexing just means that you're combining multiple primers into a single reaction. So these are the exact primers that you'll see in the module, and the exact size ranges that they're predicted at, etc. This is all relevant to the activity that you'll be doing. So we have two reactions, reaction A and reaction B, and I've used 3 different types of dyes attached to each of these primers so that they'll fluoresce in the reaction. We have our hex, 6-FAM, and ROX. Hex is greenish, 6-FAM is bluish, ROX is reddish. These are the amplicon sizes, so that means that the primer that's dyed with this hex dye is going to express in the amplicon is going to be in the 265 to 289 range, 195 to 213, 131 to 160, etc. And the reason why I sort of doubled up, I made it so that each primer was a different color and each primer existed in a different size range. You can use different primers in the same size range as long as they have different colors, but In this case, it just made it simple to do this. Then I perform my PCR, and then we set the data off for sequencing. So now I'm going to jump over to a video of me doing the actual PCR, or setting up the PCR reaction, so that you can get an idea of what the methods for this looked like. All right. Very sticky. So this is played in hyperspeed because this process takes about an hour. So what I'm doing is I'm setting out my forward and reverse primers for each of my primers. So each of my microsatellite loci have forward and reverse primers. Setting them up here, and then the first thing I'm doing is I'm setting out all of my samples and I'm writing down where they are in my plate. Now in this class, and if we were doing this module in person, we would be using eight tube strip tubes for each person. In my case, doing this in bulk and wanting to get it done quickly, I use 96 well plates. And so this is what I've printed out to basically map each of my samples. These are samples sitting on ice so that they stay cool, but not frozen. And while I'm filling that sheet out, I'll talk a little bit more about some things on my bench. This is a mini centrifuge, so it's just to basically get your liquids down to the base. It's not for any particular reactions because it has a relatively low rate of rotation. I have a little waste bucket here. I have a little cooler that has all of my reagents in it, pipette tips, and now I'm moving on to making my master mixes. So I talked about the fact that I have two reactions happening. So I have A and B, so the master mixes for A and B are going to be different because they're going to contain different primers. Everything else about what's contained in those master mixes is the same except for the primers. And the other thing that I didn't mention before is that these reactions actually have different annealing temperatures for the different primers that are contained in them. So that's another reason for doing these two separate reactions. So what I'm doing here, I believe, is making my master mixes. So I'm just adding very small volumes of each of my primers to these master mixes. I don't have it on hand, but it's a very small amount of master mix that actually goes into the PCR. Making more master mix. Almost done there. You see that I have a lot of different tip sizes depending on which reagent I'm using. This is something that's standard that we've talked about before, using the appropriate pipette tip based on the volume that you're pipetting. Oh, and then also here, this is my protocol laid out that with the white balance is hard to see. And then I've also pre-labeled each of my, plate covers based on which reaction is happening. Because as I said, these are done at different temperatures. They're going into different thermal cyclers. I want to make sure that I'm really consistent in who's getting what. And so here are the appearance of our 8 tube strip tubes, and these are where I'm going to distribute my master mix into, because I'm going to take it out of there using a multi-channel pipette and distribute it across my 96 well plates. So I'm labeling my tubes, or my plates. And then this is a really, really neat pipetter. This is, it's electronic, it's ergonomic, it's a 12.5 microliter pipette from Integra, which allows me to very easily pre-program it to say you're going to take up this much liquid, deposit it into here, mix it X number of times. So there I am distributing my master mix, and I'm distributing my master mix A to my A plate and my master mix B to my B plate, of course. And then I'm checking my plate to make sure that all of my wells have the master mix in it, a very important step. And then I'm setting up my A and B, and you can actually very faintly see the A and B written in Sharpie on the lids of each of these tube boxes that I've then turned into plate holders. So what I have now is I have all of my samples, and the reason that I have them in every other row isn't because one is A and one is B, it's just because when you open the tubes it's easier to have a space in between each set. So if we were doing this lab in person, you would be doing essentially one row of this, which would be a mother, the potential fathers, and then a few of her offspring. So once I'm done with the plate, move it to the side. if there's any errors along the way, like the pipette doesn't take up a sample, that's why you'll see me, I always check my tips and make sure that there's actually liquid in each of my tips. That's something that you have to do with, that's that quick movement. With whenever you use a multi-channel pipette, there's always something that could go wrong with one of your channels. So checking, and now most of the rest of this is just going to be me distributing my DNA samples across each of the wells. And so I'm going to also, something also to note, is that the same DNA sample is going to be spread across one time in here and one time in here, because that same sample is experiencing the reaction both times at the different temperatures with the different primers. And every now and then you'll see me go in with a single tip, and the reason why I'm going in with a single is because something happened and went wrong with the multi-channel. And that's, let's see, I guess other things to note here. It looks like an Integra ad. I have it in the corner there. I didn't mean to have the Integra box in the corner. But I also have all of these, so this is kind of a quirk of the building, but we don't have a good ice machine in my lab. And so all of these have a frozen block underneath them. So most reactions need to be done cold, especially with PCRs like these. We often have some sort of hot start polymerase in there, and so you don't want your samples to get too warm because you don't want the reaction to start until it gets into the thermocycler. So all of these have ice underneath them. This one does too. This is a really cool, basically a desk freezer. It'll keep your samples cold for an incredibly long time, and actually frozen for an incredibly long time. And so that's why it's okay that all of my reagents are sitting in here, because they're nice and chilled. And, you know, to give you an idea, each of these are 96 well plates, and so I'm working with 96 samples right now. They were all extracted using a pretty standard DNA extraction protocol, of which we've talked about a few times in this course. It's not too much longer. These are really nice tube cap openers. These ensure that you're not getting your finger on the rim of your sample when you're opening it. But for the most part, PCR reactions are fairly simple. And so this video itself is about 8 minutes long, but it took, I think it might have taken a little bit over an hour in person, and that's with me working very quickly. And as you can see, I'm very focused, which when I do this normally, and maybe I'm a little bit slower, I just wanted to make it look good for the video. The other thing that you might notice about this pipette is that the distance between these tubes is not the same distance that there is between those plates. That's another reason why I use this pipette. It can actually adjust the distance between the pipette tips. It's really neat. So then there I am, my reactions are done. I'm covering my plates. I'm going to take a little bit and make sure that I seal them properly, make sure that every single well has, as one of my advisors once told me, a thumbprint over it, because something that definitely happens and is really a shame is when one of your samples dries out in the thermocycler. So there was the video of me doing my multiplex PCR. And once that is done for all of the samples that I need it to be done for, which is quite a lot, then I send them off for sequencing.